Okay, well, thank you for the opportunity to give this extended tutorial, so 2 parts, very luxurious. And so in the second part, I'm going to focus in on spatial aspects of tumor evolution. On the large scale, metastasis, cells migrating between distant anatomical locations, and then on the micro scale as well. Metastasis, I mean, this is like one of the key things to understand in cancer, because really metastasis is the real killer. And it's estimated to be responsible for like 90% of the deaths. So sort of understanding when and how cells leave the primary tumor during this evolutionary process, migrate to some distant anatomical location, and start a new tumor is one of the big questions. And a sort of simple version of this question is we could actually just sort of ask whether a metastasis, a tumor at a distant location, was a result of a single migration event, or whether that metastasis might have been a result of multiple migrations. So this is sometimes called a monoclonal metastasis versus a polyclonal metastasis. It has an origin that has from multiple distinct cells. And the challenge here is that first, to even ask this question, we have to get data from an individual that has a primary tumor and multiple metastases. That's difficult. And most of the studies that I've worked on where we've done this and others are due to rapid autopsy programs. So patients have passed away from this metastatic cancer. So I'll show you some where there's multiple metastases. So it's the patients donating to science for the benefit of some of the discoveries that we try to do to help future patients. Okay, so we need to sequence a primary tumor and multiple mets from one individual, and then we need to build the tree. I talked about that in the last section. I'll assume we've done that. But then now we still have a new problem because we don't know when we sequence at one time from this primary and mets where these migrations were. So we still have to infer the past, but we have to infer the past for just this idea of cellular location.
Last question, ready? So you're not really, the issue is not really a single migration or multiple. It's really whether there's a single clone or multiple clones.
Yeah.
You're going to use sequencing to determine that.
Exactly, yeah.
So if there are many cells from the same clone,
yeah.
we
wouldn't see if they were, yeah, assuming the clone was genetically identical, yes. Yes, there's always this resolution issue, meaning like how many mutations can we measure to distinguish?
Just make sure I understand what you mean.
Yes.
Multiple migration means
yes.
from
genetically distinct different cells. Yeah. Okay, so these studies started a little over a decade ago. Just to show you kind of one example of this, here's 3 patients with prostate tumors. Each of these arrows, so the tumors in the prostate, each of these arrows is indicating an inferred migration between different metastases from this individual. So it metastasized to the lung, to various organs down here is another patient. So you can see these arrows, there's lots of inferred migrations between these metastases. Sort of a different representation where they're kind of showing this sort of fat tree where there's the genetic distinction, and then there's like the different clones with these little colored bars. But again, you have this idea that the liver here is a result of like multiple migrations of different genetically distinct cells, and here in ovarian cancer. So lots of like this polyclonal or multiclonal seeding. And so a few years ago, we were looking at these papers and others, and we sort of interested in how these inferences were made. And so going back again to our simplest model. So the simplest model we have is actually the same model as I showed you in the last section, but only now we have one character of interest, one thing. That character is the anatomical location of the cell that we measured at the present time. So here I have it as a color. So maybe green is that cell was from the primary tumor, and red and blue were like different metastases, right? So I built out the tree using some of the techniques I described last time, and then we now have the location of each of the cells. But we don't know the location of the ancestors, right? And that's what we need to infer, and that's what will determine how many migrations there are. So I could try to find now a most likely ancestral reconstruction. So I label each ancestral vertex in the tree by a color, and I try to do that in a way to somehow maximize the probabilities of the transitions and these branch lengths. So it's the same model as last time. Now the trick, though, is what's this sort of probability? This is now a probability of a migration. When I change state, I change color, a cell migrates, all right? So what's the probability that a cell should migrate from the primary tumor to, if red is the liver? You know, that would be the parameters of this model.
So is the first tree up there just constructed using the genotypes of the
Yes,
different
populations?
cells?
Yes, we would construct it using the genotypes of the populations, using the techniques from the first half. And now we have this new information. We have the anatomical location of each thing we measured. Okay, so we could attempt to write down this probability model. I'm going to simplify this model, but what I'll show you works for the more complicated model. I'm just going to have a simple score where if the cell doesn't move, it scores 0, and if it moves, I count that as a migration. Okay, so there's either a migration, here there's no migration, here it migrated, right? So I count the number of migrations. So this is turning it into a sometimes called maximum parsimony model. Maximum parsimony minimizes the number of events. So here, minimizing the number of migrations. So the problem is a very straightforward one. I teach this to my undergrads. Next week, in fact, I'll teach this. I've given this tree with a color on each leaf. Find me a coloring of each ancestral node that minimizes the number of color changes on the tree. Okay? This is sometimes called the small parsimony problem. It's solved in a very simple way because it's a very simple recurrence. You look at any node in the tree, and I can compute what's the minimum number of migrations in that subtree, and then I ask for another node to find the minimum number. I just look at the sum of the minimum numbers to each of its children. And so I can start at the bottom of the tree, and I can go all the way up, just counting the number of migrations with this recurrence. I turn it around. This is called dynamic programming, right? So very straightforward, very easy. And so now when we've done this labeling, we can then go look at what were the changes that happened. So here, it stayed in the primary tumor, if that's green, and then it migrated here, 4 migrations. You can see those 4. And I can actually just summarize which migrations happened. And I'm going to summarize them in this directed graph. Okay, so I'm just going to build this graph that just summarizes the transitions that I observed on this labeling. So here, the primary tumor and 2 different metastases. Each time there was a color change, I added a directed arrow. So the total number of edges here is 8 migrations, and you can kind of then start reasoning about this. You say, oh, look, in this labeling, cells left the primary tumor and went to this metastasis. Some other time, they came back to the primary tumor. They circulated back and forth between these 2 mets. Okay? So it's just a summary of this labeling. But now these summaries are what the biologists are talking about when they ask whether it's monoclonal or polyclonal or what that pattern is. Because now we can look at these graphs, call this a migration graph, we call this. It's a multi-graph because we can have multiple edges between the sites. And we can write down a taxonomy of these migration patterns. So here's sort of the simplest one, where if the graph was a tree with no multi-edges, then that's this simple case. It's monoclonal. Every anatomical site was the result of a single migration from some other location. But then more complicated things can happen. It could be directed cycles. There could be various weird things. There could be multi-edges between them. And so we can write down this whole taxonomy. And I'm not going to sort of go into all the kind of details of this. We did this a few years ago in a paper that we published.
So I'm admit to being confused.
Yeah.
So one of the things I've been interested in and various other people have been interested is, for example, a typical metastasis formed by single cells or maybe a cluster of cells.
Oh, great question.
So there's no spatial difference, right? The cluster goes from the primary to a given site, so you have no site information, but there's still a question of whether it arose from one cell or multiple cells.
Yes.
So I'm trying to understand how this maps onto that question.
Yes.
I don't get it yet, so keep going.
It's on the next slide.
Oh, next slide.
Because it has been reported It's almost like we were planted, that clusters of
that's a
cells
paper by,
can migrate simultaneously. So what would we see when we did this? Well, we might see that there are parallel edges between two sites. Because if those group of cells, now if they're genetically identical, then you can't tell.
Sure.
So you have to do it some other way. But if they were genetically different, they would be two edges between these different sites. We could detect it using it, and it would indicate that there are these parallel edges. We actually call these co-migrations in the paper, and we actually accounted for that. And that was one of the interesting things in the paper. But the other thing is that you could have other stuff that happens. So maybe a cell coming back to a location where there's already a tumor could be more likely. This is sometimes called the seed and soil hypothesis in metastasis. Like once the anatomical location is primed to be ready for a tumor cell, that more can come there. This is an old idea. It could be less likely though, because once you have already had a metastasis grow to a certain location, so liver might grow out to a big tumor, and then if another cell comes in later to that same liver metastasis, it would have to grow to enough cells that we would measure it, so we might not see those things. So there's all, you can imagine that there's all these different considerations. And so these different considerations would give very complicated models for how we should do this labeling. Because now we can't do it in the simple way where we count migrations. We'd have to perhaps change the probabilities in a dynamic way according to which migrations have already happened. People can do that. It would be a very complicated phylogenetic model. I've never seen anyone write down this model yet, but it could be done. We did something much simpler to try to approximate this. What we did is we actually looked at these summary graphs, and we asked another question, which was, could you label the nodes of this tree in a way such that when you look at the summary graph, it is a tree? So nothing ever comes back where it was before. It doesn't have any migrations coming back. Or you could do it in such a way so that it has the maximum number of multi-edges if you wanted to analyze this idea of clonal metastases. So now you can try to say, all right, here's all the directed graphs I could have as the summary. Let's constrain this so that I only look at the ones that are trees. And let me try to find labelings so that the summary graph is a tree.
So you just mean there are no loops, but you're
No
still
loops.
allowed multiple edges?
You could still have multiple edges. I could ask the one for
Sort of a multi-tree.
a multi-tree, yeah. I
Or
could
a
do the
weighted
multi-tree
tree.
version. I could do the version where it's there's no directed cycles. So that would be one where something never comes back to where it was before, but I could have a metastasis that was seeded from two different sites. I could do anything like along this taxonomy that I wanted.
So you're always looking at directed graphs, right?
Yeah, because directed is like for the migration from one to
Sure.
the other. Yep.
So, sorry, I've kind of gotten lost between the algorithms and the biology.
Yeah.
So, you talked about clusters of cancer cells migrating, and then a paper that suggested that's happening.
Suggested that it happens, yeah.
And then on the latter part, which I think is now what you're focusing on, the seed and soil hypothesis. You didn't cite anything.
Right.
This is just a hypothesis.
Yeah, it's a hypothesis. I should have put the reference, a really old reference actually, even in the 19th century people were talking about this. So there's a whole series of work where this has been described.
Okay, so the biologists believe that that actually happens.
It's a hypothesis, right? It's a hypothesis.
Now what you're about to do is that, are you looking to try to devise tests so that you can check that hypothesis?
Yeah, ultimately, yes. Yeah. And now, this is what we want to do. We want to actually play around with this idea of, could there be evidence for seed and soil? Could there be evidence for no directed cycles? All of this is like conflated by the fact that we don't know the ancestral labeling. We're always inferring. So now what we're going to try to do here is we're going to infer with these extra constraints. And this is what we would need to do a test. So I could find you the minimum number of migrations, that'll just be an answer. But now I'd want to test, well, how does that compare to migrations if I did not allow things to come back? If I didn't have a directed cycle, then how many would that take? And then I could compare these probabilities. So this is sort of the setup for doing analysis like that. Yeah.
I have a biological question, and I'm very ignorant in this field almost completely. So I assume that specialized tissues like lung or liver basically can't grow in the wrong place. That the lung will not allow liver tissue to grow there, generally speaking.
for normal cells, yes. But cancer cells become de-differentiated in various ways.
Right. So
and
that's very important, I assume. They
That's
have to
very
be
important, yeah.
de-differentiated in order to be able to metastasize.
Exactly. And we'd love to know what were the molecular phenotypes that allowed that cell to do that, right? Like
To even take root in some other organ.
Yeah, exactly.
There are biases though, right? So in other words, there's tropism where some breast cells can grow in the lungs, they can grow in the bones, but they can't grow in the pancreas, for example. So there are biases that are known.
Yeah, because there's a whole hierarchy of cell types and differentiation and in normal development that happens.
And in the progression of a cancer, is there a point before which you don't see the cancer cells in the bloodstream, and after which they're just always there?
Hard question. I don't know the latest on ctDNA. There is certainly a debate about how early metastasis is. And there are papers in recent years from sequencing studies like this that will claim that metastasis happens very early. So like even before you can detect the tumor, it's already metastasized in some cases.
Well, I mean, it might be circulating, but not really. It hasn't found a place where it can replicate.
Yeah. I mean, there's ctDNA companies that are out there that have been examining this exact question, right? Can you detect a test? Can you, in the blood, sequence blood and determine whether or not the tumor's there very early? That's a $1 trillion question. And there's active clinical trials on this.
Since you mentioned that, just for curiosity, sorry to disrupt it.
Yeah, it's a tutorial, so we can talk about whatever
comparing lymphatic system migration to circulation. I never understood which one is more important for.
Oh, yeah. This is a good question. I don't know the answer, except that when we did these studies, but I wasn't going to show this data, there's always questions about, did metastasis first go to the lymph nodes?
Well, I'm talking about going through the lymphatic system, not necessarily just.
There's plenty of evidence that metastasis for some tumors invades the lymph nodes and then might circulate through the lymphatic system.
Right.
But
because we wouldn't be able to measure this with CTCs or ctDNA.
Yeah.
Isn't the main reason that you might use the lymphatics also because that's where all the immune cells are, and the immune cells are in the lymphatics, right? And they often can be indicators also of cancers. So you look at lymphatics a lot partially because the immune system is centralized there, and they can also help us understand. Sorry.
Yeah. No, it's fine. We'll, okay. What do I want to say here? Yeah.
Can I have one more? So,
Yes, please.
So you have you want to find the tree, and then you have.
Oh, good. It's a question about this stuff. All right, great. Yeah, I'm happy to get a question about what I'm actually talking about. Good.
So you have some rates of this metastasis formation and the metastasis moving.
Yeah.
And I thought that depends on the size of the tumor, the size of the metastasis. Do you take that into account?
Here we don't. We could, but at the moment we've been doing everything from the trees constructed from the sequencing data. But, and this would be a good way to inform the models further, if you have information about population sizes, so the size of each of these mets, that's certainly things that would change and inform these probabilities.
Also the relative locations. You can go downstream from something that will change the probabilities.
You could encode probabilities, and there are biases of which tumors metastasize to different sites. Yeah. So encoding that model, I would say no one has a good model for this yet, but hopefully someday there will be such a model. I mean, we need data. We need a lot of data to, I think, get at that model. We took a crack at it, but we felt like there wasn't enough data to do it reliably. Okay, so this is actually a fun problem. We can solve this problem. We did it. I'm not going to, I think, go into details because I really want to talk about sort of the second half. Let me just show you one thing about it, and then we'll move on. This was a data set from a few years ago now where they had sequenced an ovarian cancer patient, and they had sort of local metastases, cancer in the left and right ovaries, fallopian tubes, the omentum, and then distant metastases as well, appendix, et cetera. And in their original analyses, if we were to summarize the migrations that they had this very complicated pattern. The primary tumor was the right ovary, and then it made each of these migrations. So there's a summary of migrations. You can see it goes from the right ovary to the small bowel, then to the left ovary, but some other cell went directly from the right to the left. That has a total of 13 migrations. But the same 13 number of migrations we can now compute. We can compute it in different ways and get very different scenarios. So in this one, cells circulate back and forth from the right to the left ovary. Right? So this shows you the sort of ambiguity in the data, and when biologists are saying that there's reseeding of metastases, there may be no evidence for it, right? Because there's an equally valid solution from the data that doesn't have this. The one I sort of wanted to mention actually is the other game we can play is we can get rid of all the complexity. We can say, what if there's no time that a cell can migrate back to where it was before? Then it's like this tree version. And it turns out the tree version only takes one more migration, 14, compared to the other ones. So now you can start to do tests like this. Now, I wouldn't do a test based on these numbers, but you could do this with probabilities. You can ask, do you really believe that there was any complicated stuff compared to the simpler version? Right? And if you did this with probabilities, you could start to actually genuinely do hypothesis tests. And this is what we're sort of starting to do. So that's just the groundwork for it. And
Can I ask a quick question on
yeah, please.
At the bottom, the number of co-migrations?
Yes, yeah. Yeah, I was going to skip this. It's exactly this. Co-migrations is just if you count the total number of multi-edges. And so actually this one has the fewest number of multi-edges. There's only 6 total multi-edges. And so this is actually parsimonious in a different way. If you believe this clustering thing a lot, maybe this is the solution you actually want. There you go, good. Then we got a model for you. All right, so I'm not going to say more about this because I want to say something about local spatial, but I'll just say this paper was here in 2018. We were very proud of it. Kind of sat there for a while, though it's been cited a lot. It's been used. And then suddenly there was this explosion of work, which was very interesting in the past year, on the computational problem. And it's doing things in sort of more sophisticated ways. We have a version of this fast machina, but I'm going to skip what it is because I want to show something on the next part very quickly because we don't have a lot of time. So this is this idea of looking at metastatic evolution, all right? Migrations between distant sites. Now let's sort of zoom in. Now we want to look locally within one tumor, okay? And this has been a dream of mine for a long time. As you can see, the type of work we do, we are very close to the data. We're looking at the sequencing data. We're trying to make inferences from exactly that data. So we want to do all this cool stuff. We need data to do it. The data you need to do this is some sort of spatial sequencing. If you want to build out trees from mutations, and you need spatial sequencing. So we're doing all this stuff with trees, bulk, single cell. We didn't have spatial sequencing for a long time. Now we do. And there's a lot of technologies that take a tissue sample and they measure thousands, tens of thousands of locations. They measure not, unfortunately, DNA. They often measure RNA. They measure gene expression, but they do this pretty reliably, and it's a huge innovation in doing this type of work. But it's RNA, not DNA.
Is there a DNA version of this? Because this is now what everybody wants to do in RNA.
It's what everyone wants to do. There are a few things. I said slide seq. There's something called slide DNA. It's not very good yet, though, and people are working on it. But it's hard. I mean, DNA is always harder than RNA. There's one molecule, right? DNA. Nucleus. RNA, you get a lot of it, and you express it. So everyone always does RNA first.
You don't get enough from the RNA to be able to detect mutations?
Maybe, yes. And in fact, thank you. This is like such a smart audience. I don't even really need to give this talk, because you always anticipate my next slides. And so why don't we go infer, and instead of individual mutations, we're going to infer these large copy number changes. So if you delete a whole chromosome, you tend to express fewer of the genes on that chromosome. Okay? So you can look at how much gene is being expressed along the parts of the genome, and you can identify over or under expression, and you can try to predict copy number changes. So these are these big scale mutations. So that's what we did. It's tricky because, of course, gene expression changes due to regulation, not just due to copy number changes. So it's tricky. We did a paper, we did this, we developed these copy number profiles now Then we can build the trees from the copy number profiles. That was the part of the first talk that I skipped because you had so many good questions. But now we can layer those onto space. And so now, from data, this was a liver metastasis of a colorectal tumor. We do a lot of inference. We infer that there are 3 different clones or groups of cells that each have a distinct copy number profile. This is a code for how many copies they have of each of these regions of the chromosomes. So chromosomes, clone. We build out the tree. You note there's only 3 clones. The tree is not that interesting with 3 clones, but we can see where some of the events are that distinguish them. And then now we can actually take this tree, though it's a very simple tree. We have the mutations. We can infer something about the rates of diffusion. This is using techniques from phylogeography. It's a Gaussian diffusion model. But it's really very simple here because the tree's only 3. But as the data gets better and we have more, we can layer on the same thing. Good, yeah, question?
So what's the strategy to choose the points where you would sample? Because the colors are, of course, only done later, right?
Yeah, so here, at least for these, you take it's the whole, you slice the tumor. So you extract the tumor, you slice it, maybe a pathologist looks at it first to make sure that there's some cancer cells there, and then you throw it on the sequencer.
Okay, but if you do spatial sequencing,
yeah,
you could still kind of make many decisions how to do this, like maximal distance, but then you're close to the normal tissue.
We sequence the whole 2D slice.
Okay.
The whole 2D slice.
Okay.
Yeah. Now, there's limitations here. It's 6 millimeter by 6 millimeter that we can sequence. If it's a bigger tumor, you have to choose, but a 2D slice of a whole tumor. Okay, so then we can do this now. So we can now go back in time, and we can infer how the tumor is growing and where it's migrating, because we can layer on genetic space, mutations, and physical space. Okay, so let me show you one example of this. Actually, we can do this in 3D, but I'll skip this. This is sort of a fun example from a paper. They took a whole cancerous prostate. So here was the whole prostate from an individual with prostate cancer. They took multiple sections from different regions of the prostate where there was identified to be cancer. They sequenced it. So we've got 5 different samples here. We analyze them all together. We infer that there are 5 different clones. These locations are the genetically distinct clones that we've identified. The light gray here was normal cells. So you can see the pattern. You can see that there's some consistency spatially with where these clones are. They're unique events, according to this tree, that are distinguishing these locations. And we can see now we can infer how it's growing out. And what was interesting here is we see this bifurcation. Here's the root of our tree. Tree bifurcates in physical space on the 2 halves of the prostate. And that spatial bifurcation is exactly consistent with the genetic bifurcation in the clones. So sort of unique events happening in this spatial evolution of the prostate that we can infer in this case.
So what happened at the black square?
Here?
Was that cancerous tissue?
Yeah, so I mean, there's a few things. We'd have to go look. There's probably not cancerous tissue exactly here. We're restricted by what they sampled. And I should say that these locations are somewhat approximate, they're from the paper. We didn't do this study, so they're sort of our best guess from their paper as to where they array these out. They had some information about it. So, this is not I wouldn't,
But that's the inferred route.
This is the inferred route. This is the inferred route, yeah. So we're using
we
the
don't
XY
know.
locations of all of these tissues, but where exactly we place this could be slightly different. This is the inferred route. Everything here is the inferred locations of each of them.
Right. And what's the meaning of the purple being disconnected?
Great question. Could be a problem with our sampling. Could be, and I'm going to skip a bunch of stuff that I really wanted to talk about, but I had so many good questions. It could be that we sample in 2D, and in fact, of course, the tumor's 3D. I just wanted to show you one example of that because we've done a lot of 3D tumor atlas building now. And in one of the examples is a breast tumor. So here were 5 consecutive slices that were built out with a spatial transcriptomics. We have another method using optimal transport to align them. There's going to be an optimal transport talk sometime this week. And here, you can see actually when we sequence this slice, there are 2 distinct parts of the tumor that are physically quite distant. But in the next slice, they're actually next to each other. The tumor is sort of branching out in 3D space. So maybe that's what happened here, or maybe it was an incorrect
And
image.
how does that impact the diffusion equations that you're doing?
So our diffusion equation that we did is currently in stuff I'm skipping, sorry. It could be in 2D or 3D. So we've actually done it in 3D when we have consecutive slices. So we did actually the same version. We had 2 slices. We can do the diffusion in 2D, and we can infer, again, with that the ancestor here is the red, and it's sort of somewhere in between these two, and it's kind of branching out into the blue and the green. So you can do the diffusion in 3D. You're just often limited by having 2D slices from this technology at characteristic
So I guess I'm
spacing.
confused by the use of the word. So you don't mean diffusion in the physical sense?
No, I mean physical diffusion.
The tumor cells are not moving; they're growing.
Yeah.
So
Sorry,
in other words,
it's
this thing here, and it's grown out and changed and genome,
yeah.
and a new clone took over.
Right.
So you're not talking about a diffusion process. That's why the word diffusion is,
it's
at least in my sense, is confusing.
yeah. Okay, it's modeled as all we have is the genetically distinct clones at the earlier time, right? And then there's a certain number of changes that have happened, and along those changes, we model like where. Well, it goes the other way. If you have the XY, you have the genetic differences here, you have the ancestor, you know how far in mutational time this is, and so you're inferring in physical space back where that ancestor would be if physical migration is correlated with genetic distance. Now, it's not migration as in that cell is migrating, it's like its ancestor was somewhere, it grew, and
That's
its
why you're using
descendants.
diffusion in a different language than physicists would use the word diffusion.
Yeah.
Yeah.
This is the way like phylogeneticists
Yeah, diffusion.
kind of use diffusion.
Your lecture is the
So I understand.
There's fine tissue.
Because they often think about like where the ancestor was sampled and where it ends
No, I
up.
understand. It's diffusion in genome space.
So the beautiful torus that you have, was that a cartoon?
Which one?
You had when you looked at the 3D in the breast cancer thing. Yeah, and on the right, there was this.
This was a cartoon.
It's just, that's a cartoon.
This is a cartoon. This is the real data.
Okay. But you're not sure, I see, so I don't see them connected there.
Right here. Now the point I was trying to make here was that this is a slice. These are tumor cells in this 2D slice. They're physically separated. Right. In the adjacent slice, they're actually kind of right next to each other. Right. So we would sort of view this as kind of more of a contiguous, contiguous tumor. So probably, you could, but you'd probably likely imagine that, they were together and then they, branched and grew out, right? As opposed to, yeah.
And I was just wondering if you saw any relationship between the microenvironment and the evolution of your subclonal population. I only ask because in around the autopsy we see it's sites that have immune cell signaling. There's certain clones that survive versus other sites. So system here that has serial kind of measurements.
Yes, in this study, which is published, we saw a bit of that. Yeah, there's some possible, but it's in there. There's some analysis. So there's some notion of immune cells maybe modulating a little bit this growth. It's not so great, and the reason why is so far, with what I've shown you, the genetic space that we're analyzing is very limited. It's few clones, it's few large copy number events. So here I'm just showing you 3 different clones. There's not a lot of things to correlate against like the immune environment there. So this correlations between genetic differences of different clones and what's happening in the microenvironment, we can do with a small number of clones, but you don't really get much statistical power, right? You'd want to do this with like a much richer spatial reconstruction. So that's on the to-do list, but it's difficult. I skipped one thing, but I showed you some stuff about metastasis migration. Migration locally, 3D tumor atlases, another thing we're doing on. Questions, some of these already came up, like, can we get better models for this metastatic seeding? Can we parameterize this model? What are actually the constraints? Are they clusters or they're not? We've just started to really address this question with data. Better spatial single-cell data, I think, is going to open up some of what was asked here, right? Like, can we get more realistic growth models, interactions with the microenvironment you was asked? Can we actually do these correlations? I think we need to get higher quality spatial data than what we currently have, but it's getting better. And then doing this in 3D and doing this sort of across modalities. You need DNA, maybe RNA for immune interactions. It'd be great to get some epigenetic stuff. And that's also starting to happen. So it's an interesting world for modelers. I think the modeling is going to get a lot better because the data is getting a lot better. So I'll just conclude with that thought. And thank you for all your questions. Here's the acknowledgments of some of the folks that did the work. And I'm happy to discuss further.